My name's Ellen, and I'm also in Professor Hunju's lab, as Claire is, and also co-advise with Professor Deborah Luckman. So today I will be presenting more on the research I've been doing for the past year. It involves designing a synthetic N-Cadherin matrix that modulates neurodifferentiated stem cells to form functional neuromuscular junctions. So there are many different types of neuromuscular diseases, actually more than 50 of them. And there are many types. The types include ALS, which affects the motor neurons, myasthenia gravis, which is an autoimmune system that affects the neuromuscular junction, and muscular dystrophy, which affects the muscle tissues. So the national cost for neuromuscular disease is extremely high. And for just one of the neuromuscular diseases, ALS alone, it's estimated that the national cost is around $1 billion. And with one individual, they would spend around $50,000 a year on treatment. And the scariest part is that many of the diseases have little to no cure. So in this case, there's a dire need to be able to build a neuromuscular junction in vitro. So these engineered neuromuscular junctions, they can be used in laboratories to screen for drugs for neuromuscular diseases, as well as for fundamental studies on understanding the neuromuscular junction. And in the clinics, they can be used for tissue regeneration and repair. So neuromuscular junction is composed of the motor neurons and the muscle tissues. And they are separated by a gap, which is why they're called a neuromuscular junction. So success in engineering neuromuscular junctions, or NMJs, greatly depends on the capability to generate motor neurons that can innervate muscle tissue and activate contractions. So unlike Claire, who's looking more at muscles, I'm looking more at motor differentiating stem cells into motor neurons. So there are many factors that affect the differentiation of stem cells into motor neurons, and one of them that plays a really big role is cadherins. So cadherins are these cell-cell adhesion proteins. They're composed of 5 ectodomains, a transmembrane domain, and a cytosolic domain. So past research have shown that with neurocells, there's an increase in the presence of N-Cadherins, which are these types of cadherins. And so there would be quiescent neurostem cells and active neurostem cells, in which case for quiescent neurostem cells, they are not dividing or proliferating, and for active neurostem cells, they are. So the research has also found that with a decrease in cadherin, there was an increase in the amount of active neurostem cells, and a decrease in the quiescent neurostem cells. Whereas when there is an increase in cadherins, there was a decrease in active neurostem cells, and consequently an increase in quiescent neurostem cells. So this brings us to my research goal, and that's to develop a synthetic N-Cadherin matrix that modulates neurodifferentiated stem cells, so we can use these differentiated stem cells to form neuromuscular junctions. And in parallel, to align with the goal of IGERT, I will be looking into elucidating the relationship between matrix stiffness and the presence of N-Cadherins on the neurodifferentiation of the stem cells. So previous studies have shown that cells would adhere to the extracellular matrix through specific bonds between cellular integrins and cell adhesion ligands, in particular with cadherins. So the neurodifferentiation level greatly depends on the number of cadherins. And so we decided that culturing cells onto hydrogels that mimicked the extracellular matrix environment would allow us to selectively control cadherin interactions. So this brings us to the scheme, to my hypothesis. The number of cadherins conjugated to a hydrogel with similar stiffness to the brain tissue would regulate the neurodifferentiation level of stem cells to motor neuron cells. And then using these motor neuron cells, when we co-culture them to pre-made muscle sheet, we could successfully build a functional neuromuscular junction. So this is where, for this first step, we would assemble the matrix, do cell culture and mechanistic studies. So we chose a biotinylated alginate gel system. Alginate because it's biocompatible, it's easy to change the stiffness of, and also it can be used for 3D cell culture. So we chose to biotinylate the alginate because biotin Avidin is a very strong bond that we can use to further conjugate other proteins on top of the gel, which is how we decided to coat the surface of the biotinylated alginate gel with Avidin, and later conjugate biotinylated N-Cadherin onto the Avidin-bounded biotinylated alginate gel. So as you can see, there's actually 2 active domains. I don't know if you remember, but in the beginning I said that most cadherins have 5 active domains, but we are using 2. And it's actually very interesting because our lab has found that with the 2 active domains, so the fragmented N-Cadherins, respond in very similar ways to a full-length N-Cadherin. And that's very good because they are a lot easier to synthesize, the fragmented ones. So for the rest of the experiments, I would be using fragmented N-Cadherins. Then I would culture cells on top of this matrix, so specifically bone marrow stromal cells, BMSCs, because they have been shown to be able to differentiate into very neuron-like cells. And then in alignment with IGRT goals, I'll be doing mechanistic studies with traction force measurements on the effects of cadherins and on the BMSCs. So traction force measurements is essentially where we have fluorescent beads encoded into the gel. And when we have a cell on top of it, they would, any of the proteins that are on the gels would sort of sense the tractions, and when we lyse the cells, the beads would move, and we can see how the movement translates to traction force measurements. So the next step would be to differentiate the stem cells. So we would be using a differentiation media that has growth supplements that are favored towards neurodifferentiation. And also to sort of assist the neurodifferentiation of stem cells, we will be culturing them on gels with specific stiffnesses, primarily stiffnesses related to brain tissue, because that has been shown to sort of help with neurodifferentiation of stem cells. Finally, the third step would be to co-culture the system. So we would have a muscle sheet, which is formed from inducing myogenic differentiation on skeletal myoblast precursor cells. We would have a hole in this muscle sheet where we would put the biotinylated gel disc with the BMSCs attached. And in the system, we would start differentiating the BMSCs into motor neurons. And we would test the functional analysis of the neurons by adding glutamic acid, which would stimulate muscle contractions. And to sort of test the functionality, we will be measuring acetylcholine and calcium. So here I'll talk about the results I've had and what I've done. So first of all, I prepared biotinylated alginate. So this was from one of the papers that was published in 2004 in Biomolecular Molecules. So what we have is that we have alginate with carboxylic acid groups, and then we have biotin with amine groups. So we would use EDC NHS reaction to sort of conjugate the 2 together, and we would form biotinylated alginate. So we confirmed the structure of the biotinylated alginate with NMR. And we tried it with 20% conjugation and 5% conjugation, and we decided 5% conjugation works biotin works the best, so that's what I'll be using for the rest of the experiments. So then I prepared biotinylated alginate gels, and I do this by having alginate and adipic acid dihydroxide, which is a cross-linker. And I used EDC hub to have a covalently bound alginate hydrogel. So we would create this gel disc later on with this system. Then I also coated the biotinylated alginate gels with Avidin. So actually, this is one really interesting thing that we found was, well, we found that we had very poor conjugation of Avidin, which is very strange to us considering how Avidin and biotin bonds are really strong. So, and what's really interesting, even more interesting to us, is that we decided maybe if we sort of combine the space between the gel and like where the Avidin can be by using a cover slip of a certain distance away, it might help with conjugation, and that's sort of what we found. So we had fluorescently tagged Avidin, and we used sort of a fluorescent plate reader, and we sort of saw the fluorescence of like gel cover slips at different distances result in different conjugation of biotin to Avidin. So that was really interesting. But anyways, we solved the issue about how to conjugate the Avidin to the biotinylated alginate gel. Afterwards, I confirmed that cells do attach on these gel constructs, and this is only 30 minutes, which is why we don't see spreading yet. But with cadherin here, we see that cells do attach, but without cadherin, we don't see any cell attachment. So this is where we started running into issues. So before, a lot of papers have shown that BMSCs can differentiate into neurocells. And we even saw this when, this is just the BMSCs on a glass substrate, and I put in neurodifferentiation media, and after 10 days, we see these very neuron-like structures. But the only problem is that when we asked our collaborators to help us with patch clamp analysis, which sort of tests whether or not there's sodium channels and if it actually works, we found that it didn't. So this is what it's supposed to look like. There's like a dip, and then all of a sudden a rise in sodium, but we didn't see any. So this is where we decided that maybe we should try other stem cells. So for example, neurostem cells or embryonic stem cells. So that leads to future work, which includes controlling the cadherin cell density, and then sort of adding the cells and seeing neurodifferentiation with neurostem cells as opposed to BMSCs. And then co-culturing the neurons with muscle tissue and testing further functionality. And with that, I would like to thank my funding sources, so NSF, IGAR, EBIX, my advisors, Professor Kong and Lekman, as well as the people in the Kong and Lekman's lab and my collaborators. Thank you.